Biochemistry theory and background. Proteins make up one of the 4 biomolecules and are vitally important in creating and maintaining a vast array of bodily functions and structures without which human life cannot exist. The structures of proteins greatly informs the use and purpose of that protein molecule, and these structures can be presented in 4 levels. The primary structure is the building block of the proteins themselves. It is comprised of a linear chain of amino acid sequence held together by peptide bonds. The side chains of these individual amino acids and their chemical properties, as well as their order in the chain, is what ultimately determines the function of the final protein. The primary structure serves as the basis and most important part of the protein. However, for the protein to be functional, subsequent structures must be added. Hydrogen bonding between these amino acids will occur and is dictated by the side chains of the amino acids in the chain. This will cause a linear amino acid chain to fold in either alpha helices or beta sheets. This bonding is what makes up the secondary protein structure. The tertiary structure of the protein determines 3-dimensional conformation and hydrophilicity on the inner and outer sides of the structure, and quaternary structures provide domains where substrate binding or another chemical or physical tasks may occur. Protein denaturation is the process of breaking the hydrogen bonds of the supporting structures, causing a destruction of the functionality of that protein, in this case, the egg protein, and this will be discussed further in the report. The aim of this experiment is to determine the effectiveness of certain chemical substances and physical conditions upon denaturation of egg protein. The hypothesis is that after 90% ethanol, boiling hot water will be most effective at protein denaturation, followed by 40% ethanol, then citric acid, and finally the 66% citric acid dilution. The equipment is 150 ml of methylated spirits, 90% ethanol, 150 ml of vodka, 40% ethanol, 250 ml of lemon juice, citric acid, 200 ml of water, 6 times raw eggs, 6 times small plastic container, and a kettle. The considerations are that the experiment should be conducted in a well-ventilated area due to the use of methylated spirits. Method: measure 150 ml of methylated spirits into a container. Measure 150 ml of 40% ethanol vodka into a container. Measure 150 ml of citric acid into a container. Measure 100 ml of citric acid into a container. Into this container, add 50 ml of water to create a dilution. Label the 6 plastic containers as control, 90% ethanol, 40% ethanol, citric acid, 66% citric acid, and boiling hot water. Crack 1 egg into each of the 6 small plastic containers. Add the corresponding substance to each of the containers except for the boiling hot water container. Also, no substances added into the control either. Boil 150 ml of water in a kettle. Pour boiling water into corresponding container. Wait 1 hour to record results for the tests. The results show that 90% ethanol was best at denaturing the proteins, followed closely by the use of boiling water, and then followed by 40% ethanol, citric acid, and finally 66% citric acid dilution. The control showed that the raw egg remained completely unchanged. The 40% ethanol had a moderate effect of denaturation. Results could be seen after 5 minutes of application of the vodka, with denaturation continuing for another 40 minutes after. The 90% ethanol had extensive amounts of denaturation of the protein. Not only did the egg white denaturate, but also the yolk toughened in its texture. Immediate results could be seen, where as soon as the methylated spirits made contact with the egg, white streaks emerged. Denaturation continued for 50 minutes after application. 66% citric acid had very minimal denaturation on the egg protein, with only some cloudy white streaks being visible. Visible denaturation manifested 20 minutes after application, where denaturation progressed for another 15 minutes. Citric acid had minimal to moderate denaturation, with clear, distinct white streaks through the white of the egg. Visible denaturation manifested 20 minutes after application, and denaturation progressed for 20 minutes before ceasing. Boiling hot water had a high amount of denaturation of the egg protein, with extensive white solids forming from the clear egg. Results occurred almost immediately and ceased within 5 minutes. Discussion and conclusion. The results indicate that the hypothesis was supported. Protein denaturation by 90% ethanol is most effective in this experiment, due to the way in which alcohols denature proteins compared to the way in which a change of pH or a change in temperature denatures proteins. Alcohol denaturation works by cleaving the intramolecular hydrogen bonds between the amino acid chains of the secondary and tertiary protein structures, which is what is responsible for the shape of the protein. The side chains instead form new hydrogen bonds with the added alcohol. When this occurs, the protein shape is compromised, causing it to lengthen, enlarge, and coagulate with the other denatured proteins in the egg white. The boiling water denatured the egg protein because it imparts such kinetic energy to the protein molecule that the vibrations cause the intermolecular hydrogen bonding to break. For these reasons, it is understandable that the ethanol and the boiling hot water were best at protein denaturation, as they target a more crucial binding for the protein, the hydrogen bonds. Acidic pHs target the salt bridges in the tertiary structure of the protein, which are bonds between oppositely charged electrostatically attracted molecules. The citric acid, with a pH of around 2 to 3, causes straightening of the alpha helix or beta sheet, and hence the egg protein will have a slightly changed appearance, but not as dramatic as the previous tests. In relation to bodily functions, the results can help to explain how the body can break down protein and how excessive alcohol consumption can lead to cirrhosis of the liver. In the GI tract and stomach, gastric acids cause denaturation of the protein in the food or drink that the host has consumed. Pepsin, an acidic mix of hydrochloric acid and pepsinogen with a pH of 1, is secreted by the gastric pits of the stomach to denature ingested proteins in order for the body to be able to absorb the monomer of the amino acids. Alcohol is used as a disinfectant as it can denature the structures of a bacterial cell. Now, excessive alcohol consumption can also damage human cells. This can be seen in the liver, where scar tissue and cirrhosis can occur if the liver is exposed to too much alcohol, as in the case with alcoholics. The experiment has shown the effectiveness of various substances upon protein denaturation. The order of the effectiveness of these agents are 90% ethanol, boiling hot water, 40% ethanol, citric acid, and finally 66% citric acid dilution in the order of most effective to least effective. These were tested against a suitable control for the experiment, which ensured that no exterior factors were influencing the denaturation of the egg proteins. The pathways in various molecules which take place in both the physical and chemical changes in the various denaturation processes are the factors which determine the effectiveness of these substances. Future improvements to this practical could reflect a more accurate representation of stomach pH, where hydrochloric acid is utilized rather than citric acid. This would give experimental results which are better suited to commenting upon the effect of pH on protein denaturation in the body. Another implementation that would benefit the experiment would be multiple trials of each substance testing, as well as a broader range of dilutions of each substance to identify the minimum concentration of that substance which would cause protein denaturation.